I want to welcome everybody here today, and particularly I want to introduce Pal Kazanjian, who's Professor of Internal Medicine and Chief of the Division of Infectious Diseases. He's led his division in their expansive and diverse clinical activities and leads a faculty that's extremely active in infectious disease research. Indeed, we count many of these faculty among the primary and joint faculty in the Department of Microbiology and Immunology. Pal received his MD degree from Tufts Medical School a few years ago, is that right, Pal? And completed his internship and residency at University of Chicago and ID fellowship training at Brigham and Women's in Boston, and he is board certified, thankfully, in infectious diseases. Most interestingly to us, Pal's been intensely interested in the history of medicine, and to follow his passion while not quitting his day job, I might add, he earned his PhD in history from the University of Michigan School of Literature, Science, and the Arts in 2012. His dissertation topic was on Frederick G. Novi, founding chair of the Department of Microbiology and Immunology in our medical school, which we'll hear much about him today. So to kick off the events of the next 3 days, celebrating the Milestones in Microbiology site awarded to us by the American Society for Microbiology, Dr. Kazanjian will recount the influence of Dr. Novi on early bacteriology as a result for those of us in the department on all of our lives. Dr. Kazanjian's lecture is entitled Frederick Novi, Beginnings of Bacteriology in American Medicine, 1889 to 1933. Pal?
Thank you very much, Harry. Yeah. Thank you for inviting me and sharing my knowledge on Frederick Novi with you. Dr. Novi, as you know, is an exceptional person. And what I want to impart to you today is what he did that was so distinct and unique in science, and what's the scope of his influence on all of us beyond microbiology. These are the years that Novi was active in his long career throughout Michigan. And to address these issues, I'd like to begin with Novi as a young man. He, at the very young age, had a predilection and affinity for science and became somewhat of an amateur scientist, joined microscopic clubs, and became interested in chemistry in an amateur fashion. And his interests led him to go to the University of Michigan, where he majored in chemistry and became a scientist, a chemist, a full doctorate in chemistry before he ever entered medical school. And this is just a representative example of one of Novi's notebooks when he was in his inorganic chemistry class, showing his exquisite, intricate design of instruments to solve a problem of creating a compound. And you can see the precision of his notes, his depictions of his experiments, and this led him to get a full doctorate in chemistry. The question is why he ever entered medical school, and it's because at that point in time, Michigan was transforming into a modern university where several of the professors had dual appointments in medicine. And Novi's professor in chemistry, William Prescott, actually taught at the medical school, as did other chemistry professors, such as Victor Vaughan. And it's probably that led him to go to medical school. But he relished in chemistry and science where he could create hypotheses and empirically verify the certainty of his hypotheses in the laboratory. But in medical school, he encountered something somewhat different type of lecture. And here's a representative example of one of his notes from his medical school lectures in 1890. And Novi this was right about a decade following the germ theory, was taught on the one hand that certain microbes cause diseases. But on the other hand, incongruously, he's taught about how to handle and treat inflammation. And as you can see in his notes, he's taught about using leeches and cupping and bloodletting and how to do bloodletting in pulse disease. So the theories he's being taught, on the one hand, is that disease is caused by an external agent. It's an ontogenic thing, a disease. On the other hand, he's being taught that it's an internal imbalance of poor humors, and to rectify health, one must extract the blood that's causing the excessive bad humor. Which one will he believe? How can he reconcile it? There's no experimental laboratory for him in the 1890s in medicine to put these theories to the test as there were in his science. Well, the answer serendipitously came to him from his mentor in chemistry and medicine, Victor Vaughan, and the creation of a hygienic laboratory at the University of Michigan on the central campus. Vaughan, at that point, was involved in public health, the adulterations, the chemical adulterations of milk and cheese, but he hypothesized that they should be doing more than that. There should be a bacteriologic analysis to track down epidemic diseases throughout Michigan. And unlike other hygienic laboratories in America that were being established at the time, being created in urban centers, Vaughan's idea was to create it at the university to integrate the sciences with a public health mission, a practical mission for identifying diseases and epidemics. That's why it was located in a university setting. And he got money from the state legislature to build a brand new facility with ample space, including Novy's own laboratory, which together was about 500 square feet, very ample space at the time. The problem was, where would they learn about bacteriology? There was no place in America to do so. So Vaughan and Novy traveled and studied with Koch in Europe and in the summer of 1888. And then Novy returned at that summer as a full-time to enter medical school, complete his science degree, and actually teach microbiology all at the same time. But by 1891, he had his degree in chemistry. Victor Vaughan, his mentor, had become dean of the medical school and appointed full-time faculty, which was a radical new thing in medicine, full-time faculty to teach with laboratories and without clinical practice, without competition for their time with clinical practice. So Novy was the first of a new breed of scientists, physician scientists, appointed full-time at a medical school. He had the exquisite training, the latest training in science of chemistry and microbiology, bacteriology, and he had ample space, and he had funding from the state lab to support his, to supply his reagents and also his space. So he was established as the first generation, a new breed of physician scientists, decades before the infusion of large federal funding after World War II. Well, how would he make a career for himself in science? What would he do with his newfound position? This, by incidence, was also the year he got married and was to have 5 children in the next decade. So what he did in his laboratory was something distinctly different. He used the specimens sent to him for public health purposes to do investigations, basic science investigations. Let me read you the words of Novy. This is a quote from him. And it says, "The laboratory was called into existence by the fundamental desire or demand for knowledge of hidden facts which bore upon microbes. The first object in the Institute is to carry on original investigations whereby the bounds of knowledge will be materially widened." There's no mention of practicality. He's trying to get to basic microbial biology, mechanisms of life. And here he's shown filtering extracts of bacteria to show that the products that they produce aren't all harmful. They're not all toxins that need to be nullified by antisera, such as diphtheria or tetanus. They're simply harmless products of bacteria, nitrates, nitrites, and so forth, which he would then inject into animals and show that they were harmless. It's how they live and survive. He was also interested in finding out how microbes breathe, how they respire. Here, showing his microbic respiration experiments with differential manometers and correlating oxygen consumption and CO2 production with rates of growth. Bacteria thrive with more oxygen. They grow more luxuriously. They breathe, and they form metabolism. That's how they live in nature. That was the focus of his work. But his work was also helped to expand the bounds of bacteriology beyond bacteria alone. And one of the key things about Novi that you find throughout all of his science is that when he encounters an unexpected finding, that he insists on having the autonomy and the latitude to redirect the scope and the focus and the aims of his experiment to pursue those findings. And here what he's doing is he's taking casein from milk and nucleons from milk, the phosphates from milk, trying to inject them into animals and look for antibodies. But paradoxically, he finds he kills the animals. So he aborts his original experiment and says, what's happening? Why are these animals dying? He sees organisms in the stain of the animals who die. He can't culture them out. So he invents new techniques, anaerobic jars to culture bacterium named after him, Clostridium novii, the anaerobe. By the way, on the left are illustrations of some of his instruments from his original publications. And on the right are his lantern slides that he would use of his bacteria to show and project to his classrooms presentations. And his innovations and his ability to find microbes in specimens where others were unable to led to a reputation that he was an instrumental innovator. So people who were looking for microbes and how to culture microbes throughout the world began to send him specimens from New York, abroad, and so forth. And here's an example of a specimen sent with a person from relapsing fever and a stain on the left showing the spirochete, where no one had been able to culture the microbe. Well, Novi devised a special instrument called a collidium sac, where it could be placed in the peritoneum of a rat and have infusions of protein concentrate the organisms and grown artificially. And for this, he was nicknamed Spy Novi, and that moniker stuck with him for the rest of his career. And when he was injecting these into rats, he found that some of his experimental rats died. He was unable to find out why. He couldn't find any bacteria in culture. And he postulated that one reason were that there were ultrasomes or minute particles, filterable viruses that did so. And he saved his blood until there were technologic innovations with electron microscopes, whatever, to detect these organisms. One didn't develop until after his retirement when he was in his late 80s. And he returned from his retirement when there were electron microscopes, co-cultures, and cell systems available to culture viruses. And he found the tubes of blood from these rats who had died and came back and actually was part of a team that showed that there was a virus causing the death in these rats. So he had interest in viruses as well. But what he's best known for is his ingenious and innovative experiments with protozoa and trypanosomes. And at the time, sleeping sickness and trypanosomiasis was a huge epidemic in Africa and was devastating the colonies of some of the mother countries, France and England and so forth, who created schools of tropical medicines in their metropoles to handle these diseases, because the economies of their colonies were suffering. But the blood was sent to Novi because people were unsure of what caused these illnesses. And what Novi was able to do was to create a special culture system whereby he could create and culture the trypanosomes in artificial media. And this shows some of the trypanosome forms which didn't really correlate with the forms that he saw in blood. So what he did was he had people send him insects, including tsetse flies from Africa, and he correlated the forms that he saw in his tubes, his culture systems, with the forms inside the gut of the tsetse fly, and then injected his pure cultures into animals and found the pathologic form. And so what he did is, in his system, not only did he recreate Koch's postulates, but he was able to show with a dynamic system how the protozoa developed in their various vectors and hosts, what the functions of those forms are, which were the infective forms, which were the pathologic forms, and he recreated this, simulated this natural environment in his laboratory setting and manipulated it to show what the causative organisms were. And for this work, he became widely recognized throughout the world as a towering figure in microbiology. People throughout the world, including Paul Ehrlich, wrote letters to him to try to co-opt him into looking at therapeutics for trypanosomes. Ehrlich at the time was experimenting on dyes and how dyes, the various types of arsenicals, could eliminate microbes, treat microbes. But Novi, although he appreciated that, his major interest wasn't in eradicating the microbe or attenuating it. It was to understand it. And he applauded Ehrlich for his practical applications. But he went on and continued to receive serum and specimens throughout the world to try to find the biologic behavior of microbes. And his next one that he did was looking at leishmania from specimens that were sent to him from Tunis. If a person who had fever and a large spleen, and he was able to again show the forms, the evolving forms that were in the guts of the vector, and inject those into dogs and show the pathologic forms. So what can be said of Novi and his science? Basically, that his focus was on the mechanisms of life, on expanding the scope of bacteriology beyond bacteria, and looking at the biologic behavior of these organisms in their ecologic domains, more so than the practical applications. And this is a very different view of microbiology in America at the turn of the century, where most people have depicted American microbiologists as having an interest in the practical applications of European-derived theories, making sera for diphtheria, for tetanus, and so forth. Novy's focus was on understanding bacteria and understanding their roles, the basic fundamental roles of organisms. But his job at the University of Michigan, where he spent his career, was not just the researcher, it was the educator. And this is where he established his first course in microbiology in 1889 that was based on the biologic principles. And in this course, he had an hour lecture and 5 hours of laboratory. 5 hours of laboratory daily for 3 months, 5 days a week. And his goal was, and here you can see one of his lecture notes from his 1891 course, was to have students learn not just in didactic lecture halls, but to test the theories that they heard, to hear about how microbes cause disease, and then show them how to inject pure cultures into animals and show them that microbes could cause disease and fulfill Koch's postulates. Learning by doing in the laboratory was the focus of his work. And his first textbook was in 1894, and this textbook became standard for microbiology courses in other major medical schools, Hopkins and Penn and Harvard and so forth, patterned their courses after Novy's. What did all this mean? What were the implications of this for medical education? Well, when Novy was doing all this, there were very lax standards of admission. Most students didn't graduate from even high school. Some were illiterate. And imagine trying to teach them the intricacies of science and the latest techniques of science of the day. Novy felt that by introducing and integrating science into medical education, that this elevated the quality of students. It increased the duration of years of medical school from 3 to 4. and overall improved the quality of medical education. This is Novy explaining what he thought the introduction of sciences did to medical education. And this is a lecture that he gave to the incoming first-year students in 1902, talking about the spirit of medicine, spirit of research in medicine, where one should seek the truth in the laboratory, should have a truth-searching goal, not commercial, not making money, but to seek the truth and to understand knowledge. Now you might say, how did the students receive all this? Students went to medical school for the practical knowledge of taking care of patients. How did they receive somebody like Novy, who was trying to introduce basic science to them? Did they see any utility for pure science? And the answer is, you can look at some of the notes that students wrote to him, and I'll just read one to you, one quote of a note written by a student who was a practitioner. Said, "The great Dr. Novy, never since have we been pervaded with idealism such as emanated from and surrounded that man. He taught us technique and he preached the truth, the truth." So students, practicing physicians, could share the appeal of pure science as well as people who went into laboratory research. Why? Well, it gave them a special knowledge that their forebears didn't have, their preceding doctors didn't. It helped differentiate them from other sects of medicine, homeopathy, eclecticism, and it allowed them an opportunity and a means, a vehicle to legitimize the knowledge they were being taught, to make it certain, verifiable, if you will. The students received this pure science they welcomed it. Now, I can't say that all students did. Only have the notes of the students who wrote back to Novy. And so what can be said about Novy is just that he created a space in medicine, in medical education, for the basic sciences, not just microbiology and bacteriology, but for all the basic sciences. This is a picture of Novy in his office by his laboratory, just showing how students would see him as they came to him and to seek his advice. But Novy, in addition to setting up the basic sciences, also helped establish bacteriology as an independent discipline of science. And he was one of the founding fathers of the Society of American Bacteriology in the late 1900s. And Novy felt that the pure science ideal was a critical way of providing unity and to provide cohesiveness into a field that was being splintered into various types of applications, animal husbandry, practical applications in veterinary medicine, and so forth. And Novy felt that the pure science ideal was a theme that provided cohesion to the field. And it also helped to explain nature and phenomenon in nature, decomposition of bodies, taking compound chemicals and converting them into their simpler nitrites for plant growth and so forth. And as such, it could explain nature through its special tools, just as astronomy could, using telescopes, explain the motion of the stars, or geology could explain the composition of the earth using their special tools. And what he did was help to create a cadre of scholars who were confident in the belief that if they adhered to and adopted Novy's techniques and had the discipline and the perseverance, that they too could apprehend the truths of science and discovery. And he gave an ideal to the growing discipline of bacteriology, to create it as an autonomous specialty from pathology. The original leaders of the society felt that the discipline had to be autonomous from pathology. And as such, it created a discipline, its own discipline, to be a basic science discipline in medical education. Now, one of Novy's students was Paul Decry, who is seen on the left. And Decry went on, after he obtained his doctorate with Novi, to work in the Rockefeller and then to become a popular writer. And he collaborated with Sinclair Lewis to write a famous novel called Arrowsmith. And in that novel, Lewis took Decry's idealized version of Novi as the ideal science researcher, and he applied it to society at large. But while Decry was in Novy's laboratory, he was trying to work on immunity to trypanosomes, injecting killed trypanosomes into animals, and then paradoxically he found that they died. And true to the laboratory of Novi, they had the latitude to redirect their experimental design from looking at immunity to trypanosomes to trying to find out why the animals died. And so Decry spent all of his doctorate working on the phenomenon of anaphylaxis, low blood pressure, inability to clot, and so forth. And this is a page from Decry's notebook of anaphylaxis, where he's injecting ether-extracted trypanosomes into rats. And then you can see in the margins Novy's penciled-in remarks. And Decry had these great comments about Novi. He said, in his precise, minute handwriting, Novi would write bosh and twaddle and rot in the margin of my work. He growled that every one of my conclusions was unjustified, exaggerated, untenable. But nonetheless, Decry admired Novi and upheld him as in the highest caste of medical researchers. And here's Decry writing to Novi, telling him that he's his one and only master, and that he'll be his only truth hunter ever. This is on Novy's 65th birthday. And Decry fed these notions of Novi as the idealized researcher to Sinclair Lewis as he wrote the novel Arrowsmith. And in that novel, the basic idea was that Martin Arrowsmith was the medical student who was subject to material temptations, but in his soul he wanted to search for truth and make discoveries, and that was his ideal that he followed in his mentor, Max Gottlieb. Well, Lewis patterned Max Gottlieb after the mentors of Paul Decry, Novi and actually Jacques Loeb, who was at the Rockefeller. But it was Novy's ideal and pure search for truths that was the major theme of the book that had widespread popular appeal in America. The book was a bestseller, and in large part because of the book, Lewis won a Nobel Prize in Literature, the first American to do so. And this is the movie with A.D. Anson as the character. Became a box office success with a script that was patterned after the book and a best-selling movie nominated for the Academy Award. It had huge, widespread appeal in American society and was influential to some people choosing careers in the mid-century in medicine, medical research, and science. So what have I told you about Dr. Novi in conclusion? Well, we've gone over the significance of his scientific work, what made his work distinct, and the broad implications of his work for medicine, medical education, bacteriology, American culture, and society. This is Novi in 1941, after his retirement, sitting next to his devoted wife, Grace, to whom he had been married for over 45 years. And Grace, in many ways, was his confidant. He wrote long letters to her for emotional support. She raised his 5 children, and he couldn't have done this science without the support of his wife. But another poignant view of this is that Novi in his retirement accomplished all of this well before there were large infusions of federal grants that permitted basic science work to be done at universities and medical schools. He created a life for himself in scientific medicine by being established in the hygienic laboratory, starting off with that being the source of his work and ending up with people sending him specimens from all throughout the world. So I wanted to just end by saying that in many ways, Novi was the first generation of basic scientists and established, if you will, the contours of medicine, scientific medicine as we know it today. And we are in many ways Novi's beneficiaries. So I thank you for your attention.
Thanks very much, Pal. Thanks very much. And we'll point out that Pal's book on Novi will be coming out shortly, and it's really too bad it's not available today. This is a captive audience for signing and buying. Also, we're expecting the great-great-granddaughter of Novi. Is she here, Heather? There she is. Would you stand up, Heather? Well, welcome. It's a special treat. As you know, yesterday Marty McFly, the protagonist of Back to the Future, arrived here in the future, and it's a good thing he didn't meet Frederick Novi and mess that up. Heather wouldn't be here, and probably none of the rest of us would be here. Pal, thanks very much. We'll reconvene at exactly 10 a.m. sharp, and to begin our plaque dedication ceremony. Thanks again, Pal.